Oh. Where? Projection, is that it?
If you want a real dark, stay off is usually good enough. This one right here.
Okay. Oh, it's just subtle. Okay. Okay. Good morning. We're starting the virology part of the course now. So these first few lectures are going to be an overall introduction to viruses, and then we'll get into viruses in more and more detail as we go on. Just before we start, I'm Margaret Hunt. You'll be meeting my husband Richard Hunt later on. And my office, here's my email number if you want to contact me. And my office number if you want to contact me. Feel free to come by at any time. If you want to check ahead to make sure I'm going to be in there, I've given my contact details. Feel free to ask questions in lectures, in the breaks between lectures. Come to my office, call me on the phone, send me an email, whatever. Maybe everybody's looking there. So. Okay. Okay. So, and also about the handouts and the PowerPoints, I have given you in the handouts suggested reading material, web links, etc. Those are to help you fill in any problems you're having, but you're responsible for what I cover in the lectures, and I will try and make it, if I don't have a chance to cover something in the lectures, I'll make it clear that you're expected to know that, and that should be in the handouts and the PowerPoint. There is extra material in both of them because I think it's easier to understand stuff if you've got some background. I'll try and make it clear as we go along what I expect you to know and what is there for illustrative purposes. Okay. Okay, so in this section of the course we're going to start off, as I say, with an introduction to viruses, structure and classification, and I'm going to be going through that for this lecture and the next lecture, and this will also extend into some of Monday's lecture, so try and remember to bring the same handouts on Monday. And we're going to start off with the introduction with some basic virology, going through virus life cycles, so you get some idea of the principles on which these viruses work, and then we will go into the clinical virology and actually look at the clinical pictures associated with these viruses. Okay, and so part of the reason that we're taking this approach is viruses have a very different structure, as you're doubtless aware, from bacteria and eukaryotic cells. And really to understand how we deal with viruses, how we develop antiviral approaches, how we monitor for viruses, you really need to understand something about the structure of viruses, and we'll be going into that in quite a bit of detail. They also have a different method of replication, and that same applies for that. And as I say, all of this has got implications for prevention of viral infections, diagnosis of viral infections, and treatment of viral infections. And one of the things is that, as you will see, I hope, as we go through, developing antivirals is actually a rather difficult problem. And that's because you basically have to target each virus on its own grounds. Finding broad-spectrum antivirals are a problem. Because of this, development of antivirals is expensive and treatment of virus infections is expensive, and in addition to which you'd rather not have a virus infection if you can avoid it. So a lot of the things, the approaches to these huge number of virus diseases that are around is to improve our knowledge of how they're spread, where they hang out, how they're spread, how you can inactivate them so that you don't expose yourself to them any more than is necessary, how you develop vaccines for them, and of course how you can develop drugs to them. Unfortunately, just as with the bacteria, we suffer from drug resistance in viruses, although the mechanism can be somewhat different. And so we need to know all these and understand these in order to approach the prevention. And again, that's the aim of some of this basic stuff that I'm going to be going through. It does have implications, and I'll try and point some of those out as we're going through it in the basic stuff. But as we come back to the clinical stuff, I hope you'll sort of begin to see why I stress some of this so much. One thing is we cannot go through every virus that infects humans. There are a huge number of them. Many of these are rarely diagnosed because we don't really have a good diagnosis for them, and there hasn't been too much effort to develop a huge amount of diagnostic material for many of these viruses, because if there's no way to treat them, then diagnosis is kind of for intellectual purposes only. But as we're now getting into the era where diagnosis is becoming much easier, much more readily done, much more sensitive, much more accurate, and antiviral mechanisms are becoming much more developed. That's both in terms of prevention at the public health level and in terms of antiviral agents. So as you get into the clinics, you're going to see much more emphasis on viral diagnosis than there has been in many past years. So one thing is, I cannot tell you about all of these viruses, which is probably going to relieve you if you know how many there are. I can't even tell you about all the different life cycles they use, which is also going to be a relief to you. So what we're going to do is we're going to focus on a few and try and get the basic principles, so that then when viruses emerge from out of nowhere, at least you can, I hope, be able to follow why people are doing what they're doing, why the treatments or the prevention methods are what they are, and be able to give good advice to your patients based on a solid understanding of how viruses work. And just to give you some idea, I guess this begins to show how old I am, but Dr. Mayer is even older. When I first started lecturing on this, the HIV virus hadn't been isolated. People asked me in the med school class, since there was no known human retroviral disease, why did they have to learn about it? And I pointed out this is a critical type of life cycle, and maybe, unfortunately, one day one of these viruses will turn out to be important. But any virus that has a similar life cycle, you should by now know about. Well, of course, I don't have to justify that anymore. But these are all diseases that have appeared in the US in the periods where I've been lecturing, from viruses that we didn't think were going to be a problem, either because, like West Nile, we didn't consider them to be a problem in this country, but they were elsewhere, or like hantavirus, we didn't even think that they caused this particular human disease that you're going to be hearing about from me later on. You're going to be hearing about this from me later on. SARS will be briefly mentioned, but it made major news headlines a couple of years ago. It seems to have faded at the moment. We hope it stays faded. But we had outbreaks of things like monkeypox in this country. Human metapneumovirus is a virus that we didn't realize caused a lot of childhood pneumonia a few years ago, and now we know that it does. So we keep finding viruses that are causing diseases that either emerging viruses or viruses that we previously didn't recognize were a cause. So as I say, all I can hope to do is give you enough of a grasp of virology that when new viruses come out, you can understand what CDC or the newspaper or whatever are going on about when they make some of the points that I'm going to be making. So viruses are constantly emerging, and part of that is global travel. Part of that is development going into areas where nasty bugs hung out on their own and never met us, and now we meet them. And part of it is just improved diagnosis makes us realize that we put certain diseases down to the major viruses that cause them, and then we find out once we start curing those or preventing them that there are other viruses that cause similar disease, and we still got a problem. Viruses actually quite an impact on society. They reckon to account for 50% of all absenteeism, and very few of those are ever actually diagnosed as to precisely which ones cause it. You know, you've got a gut problem or you've got a respiratory problem, you stay home a few days, nobody diagnoses it, but many of those are virally induced. And it's estimated that children get multiple visits per year to their pediatrician for viral infections. So it's a huge drag on the social, economic, and medical costs. So viral infections, just to point out that some of them are just, you feel pretty miserable for a couple of days and then you recover. And those are no big deal, except what you tend to find is some of these viruses can be major problems. Most people, they're minor problems, but in certain people with predisposing conditions, they can be major problems. So if you've got somebody with underlying heart disease, pulmonary disease, immunodeficiency, children with an immature immune system. These kind of situations, these viruses which in the average adult individual are not a problem, can be a major problem under certain circumstances. So that's where you need to know when they're going to be a problem and how you can protect those individuals at risk. They can cause persistent disease. So some viruses, once you've got them for life. And you may not be aware you've got them, and you'll hear herpes viruses are the classic group of these, and you'll be hearing about those from Dr. Gangemi. But once you've had a herpes virus infection, that herpes virus hangs around with you for life. If you're lucky, it will never cause a problem again, and if you're unfortunate, it can cause continual problems. They can cause congenital disease, and so if you're in Obgyn, this is particularly important to try and protect the fetus and the newborn from the severe consequences of some of these viruses if they're acquired in utero. They can be a contributory factor in cancer. In humans, no virus per se that we know of will cause cancer. You can be infected with some of these viruses. They increase your risk for developing cancer. So one of those are the human papillomaviruses. Even if you get the strains associated with cancer, only a small percentage of the people infected get cancer. But it turns out that virtually everybody who gets cervical cancer has been infected by one of these papillomaviruses. And so this year we now have all this big publicity because we actually have a vaccine that is based on being a vaccine for the human papillomavirus. Take one of the risk factors and you don't progress. It seems to be an essential risk factor, at least in the majority of cases of cervical cancer. So this is actually now, maybe the incidence of cervical cancer will be reduced hugely as a consequence of this new vaccine. There are several of them, actually. Different companies have come out with them. And they can be a contributory factor in other diseases. And again, you'll be hearing about some of these. But some diseases can be relatively minor, but if you get a particular virus at the same time, it can really contribute towards progression. Okay. Do stop me if you've got any questions. I only had cereal for breakfast. And also remember, a lot of these viruses are asymptomatic. Oh, the question was, I've mentioned in my notes that viruses are being implicated in juvenile diabetes. In many cases, the mechanism is not clear. One possibility is that they can actually target the islet cells in the pancreas. But another possibility is some of these viruses may have epitopes on them that cause an immune response, and that immune response can target just by sheer bad luck. Yes, and we'll be coming up to it. There seem to be some viruses that when you get them, there's a finite chance. But it's not 100% of the cases. So there appear to be some other risk factors involved. So it's not, yes, you will get this. We'll come to that when we get to some of those viruses. Some viruses have been, people with obese, in some cases, some antibodies to some viruses appear to be more frequent in the obese population. And so there's some question as to whether they may be involved in changing some kind of thing that's involved in obesity. A lot of these are very fuzzy at the moment. And part of that is it's been very difficult to find a few viruses in your body and what they're doing, because it's not ethical to infect people and find out how many go down with diabetes. So doing the experiment can be very difficult. So a lot of it is circumstantial, indirect, and trying to find traces of the virus, but it's difficult to actually isolate the virus. But with the modern techniques like polymerase chain reaction, where you can find very small amounts of material very accurately and very specifically, again, I think you're going to start finding as you get into your practice that you're going to see a lot of these viruses becoming a much more obvious how they work. A lot of it at the moment is, as I say, guilt by association, and we're not sure the association is necessarily correct. So here's the sort of positive spin for the viruses. They can be useful. Some of them infect us. We have them for their whole lives. We're never aware we had them. As far as we know, they gave us no symptoms when we got them. They can hang out with us. We can get them time and time again, some of them, and we just never know. But what we are beginning to find out is some of those are not as benign as we thought, because now we're seeing in the last 20 years, with the advent of so much transplantation, with the advent of HIV, we're seeing a lot more immunosuppressed people, and we're finding a lot of these viruses can be more of a problem in the immunosuppressed than they are in a non-immunosuppressed population. So, but viruses can also be useful. Not only do we develop vaccines against viruses, we can use viruses to develop vaccines, and we'll discuss that later on as some of that. We can use viruses as a tool to carry genes into other cells in gene therapy, and that is being heavily developed, although there have been a lot of false steps along the way, and some of the trials have been rather disappointing. But again, I think that will eventually take off quite nicely. But at the moment, it's still under clinical development. And they have been widely used as tools to investigate host cells. You probably don't care about that, but a lot of what you learnt in cell biology and biochemistry was developed using viruses, because they are very convenient ways to put bits of nucleic acid into cells and to see what happens. Okay, what are viruses? And I can come up with some very elaborate explanations which really don't fit it. A famous French virologist, who was one of the founding fathers of virology, said viruses are viruses because viruses are viruses. And it's a difficult thing. I hope by the time you get through this, you'll know what a virus is. But coming up with a good definition is difficult. And so what I think this was a New York Times reporter, and he said a virus is a piece of bad news wrapped up in a protein. And that is a pretty good definition of a virus. So let's go into what they... Let me just make sure I'm the right one. Okay, so what is the bad news? The bad news is the nucleic acid. That's what controls what a virus does, where it grows, how it grows, what effect it has on the host cell. That's all a consequence of the genome. So the genome is the real problem here. And in viruses, the genetic material can be DNA or RNA. So that's where they differ from us. Our family jewels are all DNA. Their family jewels can be either. That, in the environment, is not terribly stable. Nucleic acids are very susceptible to UV irradiation because they absorb at the same wavelength as UV light that comes through from the sun. And also, RNA is very unstable in the environment. Our skin is covered with ribonucleases, so we would just thrash it as soon as we found it. It needs to protect that genome. As I say, this is the family jewels, so you need to protect them, and so they're protected by a protein coat. So that's wrapped up in a protein. That's your basic minimalist virus. And that protein coat is also important because the viruses have to get into the cell, so they need protection from the environment, and they need a means to get into the cell. And we'll go into that in a bit more detail, but this protein coat is involved in getting them into the cell. And in some cases, the viruses actually don't just wrap themselves up in a protein. They have a lipid envelope around the outside, and they get that, by and large, from the host cell. There's one exception, which are the pox viruses. And those are the real pox viruses that cause things like smallpox. Chickenpox is not a real pox virus. It's a herpes virus. But these are the real pox viruses. And you'll be hearing about them periodically, because if there's an exception in what viruses do, it tends to be the real pox viruses. So they get this lipid envelope, and they are small, and I'll give you some ideas of the scale. And one thing is, to give you some idea of scale, I'm putting the size of these virus particles on. I'm not going to set you an exam question saying, is pox virus 20 nanometers in diameter or is it 400 nanometers? I don't care. I'm not going to ask those kind of questions. But I'm putting them there so that you can get a feel for what's small and what's large, and you can sort of put it into your own scale image. So we're going to be, when we start talking about the DNA viruses, we're going to be working up from the small ones to the large ones. So I'm just trying to give you some idea of what they are. Don't learn these numbers. And they are obligate intracellular parasites without exception. You can't give them a really good medium and grow them. They won't grow. So they all require a cell to grow in, which of course is different from bacteria. Many bacteria do not. Okay, so what about scale? Oh, well, this actually just shows how much viruses vary each from each other. These are a mixture of DNA viruses, many of which we're going to be talking about. And you can see they vary from, these are to scale, they vary from very small, parvo means very small, up to these big pox viruses. Similarly, the RNA viruses, we have things that are really small. Pico means really small as well. If you've got Latin, you're well ahead here, except they may be Greek. I'm not very good at working out which is which. But anyway, picorna stands for pico, meaning very small, and RNA meaning RNA. So you have these really small ones, and you have these really large ones. And I think if you look at these, you can see they all look very different, and it looks very confusing. But I hope by the time we've gone through these next few lectures, you'll start to see there's common principles about building viruses, and actually these are just variants on them. And once you understand them, it's like knowing that floor plans consist of, a kitchen and a bedroom and a bathroom. And once you know it, you go into a house and you look for the kitchen and the bedroom and the bathroom, and they've all got them. It's just basic building blocks. And the same thing here. This mature virus particle we call a virion. And that's kind of, we all have our jargon, but it's jargon because if I talk to you about a virus, do I mean sort of, if I talk to you about, you've got West Nile virus, that's talking about virus in that sense, virus in terms of disease transmission. If we want to talk about a virus particle, we need a specific word for it, so we call it a virion. So the mature virus particle, the thing that spreads from one individual to another, is called the virion. I said about scale, here's Staphylococcus. E. coli would be, of course, it's a rod, and it would be about 4 times as long as this. So this is Staphylococcus. Here are the chlamydia, the smallest of the bacteria, and this is an elementary body, which, of course, would swell up before it becomes active. So this is only a sort of storage form. And this is the smallest of the bacteria. And here's pox virus. So it's just about getting to the same size as chlamydia, but it has a very much smaller genome. And most of the viruses are much smaller than pox. Pox is just about the limit. With dark field illumination in a microscope, you can see a sort of point of light, just about, if you've got everything beautifully set up. So none of these can you see any details in a light microscope. You have to look in the electron microscope for all of them. Just a quick summary. I think I basically said this, but just summarizing some of the major differences between viruses and bacteria. Viruses never grow on artificial media, whereas bacteria often do. Bacteria divide by binary fission. They grow larger, they split in two. Each grows larger, it splits in two. Viruses don't do that. They disassemble, they replicate their nucleic acid, they replicate their protein, and then they assemble new virus particles. So it's a very different mode of replication. The family jewels in bacteria is DNA. It can be either in viruses. Bacteria always contain both DNA and RNA because, of course, they've got ribosomes, messenger RNAs, tRNAs, they've got the whole lot. So RNA is an important part. It's just not the genome. Viruses usually have either DNA in the virus particle or RNA, but they don't regularly package both. The arenavirus family contain protein synthesis machinery. Bacteria, of course, do. Viruses don't. They're going to rely on the host cell for protein synthesis. There's one minor exception, and don't worry about this, but people always come to me and says the textbook says this and you said not. So, the arenavirus family, arena means sand because arenas in olden days were originally had a sand floor. The arenavirus family looks sandy, and the reason it looks like it's got sand inside it in the electron microscope is because it appears to package ribosomes, but that just appears because it's sloppy, and when it packages its genome, a few ribosomes get in. They don't seem to have any function. So technically, one group of viruses does put some protein synthesis machinery, but it's only a few ribosomes. It's not going to do anything, and they don't seem to have got, as far as we're aware, any function. They just seem to be accidental passengers. Bacteria have uramic acid. They have special cell wall structures that are shared by many bacteria, so you can have a lot of antibiotics that can target these. Viruses do not. So that means broad-spectrum viral wall antibiotics don't exist. Bacteria are sensitive to a lot of antibiotics because they share a lot of common pathways that we don't, fortunately. But viruses, as I say, they tend to be, each family tends to do its own thing its own way, so you don't get broad-spectrum antibiotics. So viruses present rather different clinical problems. So the consequences of this is we don't get any broad-range antibiotics, and they're heavily parasitic on the host cell. And so the problem is that if you target viral replication, you tend to target your own cell, and you tend to die cured, which is not terribly useful. So one of the things is, as we go through these, and one thing I'm going to be stressing, we're going to go through the life cycle of some of these, choosing some typical viruses and go through the life cycles, prototypes, in order to see what kind of things they do. And one of the things that I'm going to be stressing is we need to look for when they do things on their own. Because if they do something on their own, you can target that. So if they take in an enzyme, their own enzyme, to do something, or they make their own enzyme to do something, if we can target that enzyme, then we can target the virus and not us. But if it's using all of our material, then it's very difficult to target the virus. And that's been a lot of the problems in developing viral reagents. So we're always looking for what it does that is a weak link, and the weak links are when a virus does something for itself. There are also some slightly weak links, is if it's using something that our host cells rarely use and can manage without for a short time. So those are sort of relatively weak links. But the important weak links are where a virus does something for itself. And that's why it's important to know in life cycles where viruses do their own thing and where they rely totally on the host cell. While we're giving a sort of overview of this, the host range of viruses can be extremely variable. Some viruses only infect mankind. So examples of that would be measles, smallpox. Other viruses can infect all sorts of animals. An example of that would be rabies. Some viruses can infect insects and all sorts of animals and man. An example of that would be West Nile virus, which can infect mosquitoes, a lot of species of birds, and it can also infect horses and man. So you have this huge difference, and that's going to be obviously important in terms of controlling these viruses. So knowing the host range of these viruses is very important in terms of control. So we get, as I say, we get these huge differences, but we don't have any viruses that we're aware of, and I don't expect we'll find any, that can infect both bacteria and humans. And of course, since this is a med-micro course, we're very focused on ourselves. So as far as we know, we can't catch viruses from bacteria, and I don't expect we ever will. And part of the reason for that is the viruses are very parasitic on the host cell, and as you know, bacterial cell walls, bacterial metabolism, bacterial enzymes, bacterial protein synthesis, it's all very different from human or eukaryotic biology. So what we have is that the viruses, no virus appears to be able to use both bacterial protein synthesis to make its proteins and eukaryotic protein synthesis to make its proteins. Bacteria have got special ways to get into, viruses have got special ways to get in bacteria across cell walls. Human viruses have got special ways to get in across plasma membranes because we don't have the cell walls. So we've got different problems. So the viruses don't cross that boundary, because eukaryotic and prokaryotic cells are apparently just too different for them to parasitize both. Okay. I said we have this huge difference in host cell range, and why do we do it? Well, one really important thing that we're going to be stressing is, I said viruses are wrapped up in protein. Here we have a virus coming along. It would actually be diffusing all over the place, but PowerPoint doesn't do that. Or at least this version of PowerPoint didn't do that. So this is an envelope virus. Here's the nucleic acid, or I'll introduce that later on, but here's the nucleic acid wrapped up in the protein. But this one has got an envelope. So that's a lipid membrane that it got by pushing through a lipid membrane to get out of the host cell, and it acquired the host cell membrane in the process. But in the host cell membrane is a viral protein, which is shown in grey here, and that grey protein will recognize something on the host cell. It doesn't matter what it is. It can be protein, lipid, carbohydrate. But it specifically recognizes that, and then it holds the virus in place so that the virus becomes very closely associated with the host cell plasma membrane. And now it can use whatever mechanism it's going to use, and we'll talk about that on Monday. It can now get into the host cell. So all these viruses require a protein on the outer surface which is going to recognize a receptor on the host cell, and we'll call that an attachment protein. So this is a virally coded viral attachment protein that's going to enable it to get into the cell. So if that protein only recognizes something that is on human cells, and it's not on mice cells or any other cells, it can only infect humans. An example of that is polio. The poliovirus receptor that is recognized by the poliovirus attachment protein, which is actually doesn't have an envelope, so it would be, that would be on the inner, this capsid, this protein-coded RNA or DNA. The polio attachment protein only recognizes a receptor on human cells, so it doesn't infect mice. They don't have it. But if we use genetic engineering techniques to put the human receptor in mice, you can now infect them with poliovirus. So this receptor is really important in affecting host range. But there are other things. Whether or not the replication machinery is available, some viruses need to grow in replicating cells. They might need a particular feature that they don't find in all cells. So which cells they can infect may be affected by whether they've got everything they need to replicate and make new virus particles in those cells. The ability to get out of the cell is important. If you infect a cell and you never get out, nothing's going to happen. You're not going to be able to infect another individual. So they have to be able to get out of the cell and spread. And we'll come back to some of these because some of these points will come up as we go through the viral life cycles. And of course, the host antiviral response can be really important. And in obvious cases, if the host is immune deficient, they may have major problems in responding to some of these viruses. Okay, so I've been unfortunately using terminology before I actually introduced it. My apologies there. But the virus particle is called the virion. And I'm just going to go through some of this terminology because I can't avoid using it. And the protein which coats the genome is called the capsid protein. So that's what encapsulates the nucleic acid, and it's called the capsid. It has usually some kind of symmetry, and we're going to be talking about that. And the combination of the capsid plus the genome, the nucleic acid plus the encapsulating protein, is called nucleocapsid. So actually, nucleocapsid is just a way to say bad news. That's nucleo wrapped up in a protein. That's capsid. May have an envelope. And the outermost surface will have to have an attachment protein on it. Okay. Are there any questions on that? Or? Okay. So one of the things that I need to talk about is how viruses are assembled. And as I say, despite the fact that they look so different, there are some underlying principles. And most viruses are built on symmetrical principles. And part of that reason is they're very small. They've got very small genomes. They only code for a few proteins. So if you're going to assemble things based on a few proteins, you tend to build on symmetrical principles, just like if you had a box of Lego, you build things on a pretty symmetrical principle because you've only got a few number of subunits to put together. And that's what the viruses have. So there's an underlying principle of symmetry here because they're going to use lots of copies of the same little building block because they don't have too much genome. They can't code for too many building blocks. They can actually do some quite sophisticated things with these limited number of building blocks. But underneath it, there's this principle of symmetry. And a lot of viruses are based on icosahedral symmetry. And that is based on a 20-phase solid. And let me just... Okay, so here's the 20-phase solid. There are just a limited number of these solids that have every face identical. So the icosahedron has 20 identical triangular faces. There aren't too many of these kind of so-called platonic solids. The cube is one, where every face is the same, and the icosahedron is one, and it seems that most viruses have gone for the icosahedron principle. So I'm just going to introduce you to it. I have some icosahedrons here if you want to see them and hold them, but it's difficult for you to see them from back there, but you can just make them yourself out of overhead projector sheet or whatever. So these are 20-face regular solids. If you look down on one of the points, you see a 5-fold symmetry. So you've got 5 triangles coming out from this point. If you move it, you can get some idea of the solid. Now, if I can stop it in just the right position, which is not simple with this software. No. Here you're looking down on the face of a triangle, and if I had this set up quite right, well, let me see if I can just do it a little bit better. Okay, we'll stop it. Down. Well, I'm having problems with this software. It just doesn't want to stop it where I want to stop it. Well, it's not going to work, so let me just go back and but you get the principle of what a solid is. It's got 20 faces. It has 12 of these points. These points are called vertices, and it has 12. And that's just a definition of it. It's just a mathematical solid. So if I stop it there, you can see it's got 5 triangles. So it's got a 5-fold symmetry here. If I had been able to stop it precisely here, which I couldn't on the current software, it's got a 3-fold symmetry. If you cut the cake in this way, you will get 3 identical slices. And it also has a 2-fold symmetry, because if you look at it from this angle and draw a line across here, the top half is equal to the bottom half. So that's, again, just the nature of the solid. But the principle of the 5-fold symmetry is going to be important, and so, to some extent, will be the 3-fold. I'm probably not going to discuss the 2-fold symmetry at all, because if you want to understand how viruses assemble, it's important to know that they've got this 5-fold symmetry, because when you look down on a vertex, there are 5 triangles. It's important to know there are 20 triangles, and it's important to know they're triangles, and that means they've got 3 corners. So where do we go from here? And with these viruses that use icosahedrons, the capsid is icosahedral and the nucleic acid is inside. And this is just to remind me. So here's an icosahedral nucleic capsid, and here's the nucleic acid. A bite has been taken out of this so that the nucleic acid can start to come out, but it's normally inside and buried inside. Okay, so what's the principle of building up on these? So if we look at the icosahedron, the simplest way would be to build a regular solid out of this, and you want this shell to be a solid shell, would be to put 1 protein at each of the 3 corners of all the 20 triangles. And so that's what I've shown here. Each one is going in a different color, and let me just click through these. Oops. Okay. So here we've just put the same protein molecule, or it could be a group of peptides at each corner. So every one of these balls is the same. So to reflect that, I just put them in different colors here so you can see how they're coming together at the corners, because each of these corners, these vertices, is 5-fold, so it's getting 1 of each of the 5 different colored balls. But since they're all the same protein, I'm just going to change them over so they're all the same color. Okay, so this is the simplest way you could do it. The simplest thing would be if each of these balls represented 1 polypeptide. There are a few viruses where it is that simple. And if you just had 3 of those on each face, because you've got to, if this is a symmetrical solid, you have to build it symmetrically. So every face is going to be the same. Where they come together at the top, you're going to have 5 of these balls together. And it turns out that in viruses, those tend to have very tight associations. So when you look in the microscope, you will see these as the group rather than as individuals, although microscopy is improving enormously, so you might have some idea that there's 5-fold symmetry there, because this is at one of those vertices at 5-fold symmetry. So in the microscope, you tend to see, in the electron microscope, you tend to see this as one so-called capsimer, one assembly, with maybe a suggestion of 5-fold symmetry in it, according to how good your microscopy is. And then you'll see all the others as well. So this is called a capsimer because it's a unit. It's the obvious unit when you look in the electron microscope from which this is built. Since there are 12 vertices, there will be 12 of these things. And since they have fivefold symmetry, they're called pentons because penton is penta or something, is Greek for 5. I think there were wars amongst virologists as to who learnt Greek and who learnt Latin. Nobody learns either these days. But so we have 12 of these pentons. Occasionally they're called pentamers, but that's an older term. I'm going to call them pentons. And we have 12 of these. Each one interacts with 5 others because that's the nature of the icosahedral solid. There's fivefold symmetry, so there'll be 5 of these that they interact with. And that's why they're called pentons. They interact, they have a fivefold symmetry. So I showed it here on my great overhead projector sheets. There's a clean X inside, that would be the nucleic acid. And then here is the icosahedral thing. Here I've shown those 5 things. Each face has got 3 of these subunits on. At the apex vertex, you get fivefold symmetry. There are 5 of these here, and they're interacting with 5 others. That's the simplest way you can build an icosahedron. This would not be ideal for a virus because they have to meet in the middle because basically this would just leave a hole. So actually they fill this space. And to show that, I have this model here, which is just showing the same thing, except these subunits here have been expanded so that they fill the face, so that the nucleic acid inside will be better protected. And so here's one of these pentons, and you can just see there's a suggestion that there's a fivefold symmetry to it, and it's interacting with 5 others. And they're multicolored just so that you can tell one from another. But if you were to look at these, this green one here is interacting with 5 of these here. So that's your simple virus particle. Any questions? Okay. So each of these will be a capsid. Each of these will be a penton. There are 12 pentons. Simplest virus will be made of 12 pentons. There are relatively few that are made of that, apparently because you can't get much nucleic acid inside a structure with only 12 pentons. So you need to grow this a little bit. So how do you grow it? You make it rather bigger. Well, if you move the pentons apart, which is basically what I showed there, you're going to get holes in there. So what you need to do is fill them in with some more protein. So the next simplest thing will be to fill each of these triangular faces in with 1 protein molecule on each face, or 1 subunit. It could consist of more than 1 individual protein molecule, but they come together to form this subunit or capsimer. And if you put 1 of those on each face, since there are 20 faces, you would have 20 of them. And if you look at each of these, they're interacting, because it's a triangle, they're interacting with 3 pentons. And on the sides, they're acting with 3 of these purple balls. And so this central one is interacting with a total of 6. And so it's called a hexon, because hex is Greek for 6. So what you have now is you have 12 pentons and 20 hexons. And that gives you something that's rather larger. And that's shown here. If you take this one, the central one, it's interacting with 1 penton, another hexon, that's 2, another penton is 3, another hexon is 4, another penton is 5, another hexon is 6. Okay? So that is what this shows. So here we've got same size subunits. This time all the pentons are shown as garnet. And all those hexons, 1 on each face, are shown in blue. And you can see that that's enabled us to get rather larger than we had before. So what we have 20 blue subunits because there's 1 on each face, 12 pentons still. There'll always be 12 pentons. That's the property of an icosahedron. So you can see by adding an extra hexon on each face, we have now got a considerably bigger virus particle. And most of our viruses have got at least 1 of these subunits on each face. However, that still puts some limits on size, and we have, as we said, we've got very different sized viruses. So the smallest ones look like this, the next ones look like this, but then you can keep on growing this face out. You'll still have the 12 pentons. They'll always be at the corners of the triangles, but you can fill in the triangles with more proteins. So here's adenovirus, one of the causes of, causes all sorts of things. Cystitis, diarrhea, respiratory problems. Members of the adenovirus family infect a lot of epithelial surfaces and causes some conjunctivitis is another one. So here we've got adenovirus. There are these 12 pentons here which are shown in black, and adenovirus has got attachment proteins that stick out from them. That's what's on these so-called fibers. And you've now filled the face with more of these subunits, so more capsomers. So we don't just have one hexon, we now have a lot more capsomers. And I just want to talk briefly about how those are packaged. But if I just take any one at the moment, and we'll explain why this is the case in a moment, but if I take this one, it's got 1, 2, 3, 4, 5, 6 surrounding subunits. So it is also a hexon. It's got six-fold symmetry in its interaction with its neighbours. And why do we tend to see that? And this is just a sort of, seems to be just a simple form of packing. Let me see if I can do this. Okay. What I have here, and those of you who've got, I have it here. I just have a little square box which is filled with little round tubes. And actually there's a picture of it in the PowerPoint in a second. If I just shape these tubes, they can get very poorly packed here. So if this was the surface, I've lost my pointer. Thank you. If this was the surface of your capsid, this wouldn't be very good protection because your nucleases could get through here and chew up the genome. So what you want is, you want this to be as well packed as possible. And if you just shape these and put them down, where they're well packed, and this is about as good a packing as you can get for little glass tubes, if you look at this one, it's got 1, 2, 3, 4, 5, 6 neighbours. It's just what happens. You can do it with ping pong balls, you can do it with oranges, you can do it with apples, anything that's approximately spherical. The best packing turns out to be six-fold symmetry. And there are probably mathematicians who spent ages pointing out why that is, but I'm not going to bother. So if we pack our little tubes like that, what we discover is the best packing is six-fold symmetry, and the viruses use the same thing. As I say, this is just sort of mathematical, how would you make it? It's the same principles that probably your 3-year-old knows very well if they're playing with Lego. So what we see on these bigger faces is these subunits, these capsomers, are packed with six-fold symmetry as you get bigger. So you get this six-fold symmetry, they're hexons. So what you get is, as you grow larger, you get increased numbers of hexons. So what we have in adenovirus, is 12 pentons, and you should always know the number of pentons. It's one number you have to know. And on the faces, it has 12 hexons on every one of its faces. Since there are 20 faces, that's 240 hexons. So it has a total of 252 capsomers, 12 pentons, the rest are hexons, and the number of hexons will always be a multiple of 20 because all the faces are identical. In adenovirus, the pentons are made up of different proteins than are the hexons. But in some other viruses, the pentons and the hexons are made up of the same protein. So there's no hard and fast rule about that. It just depends on the virus. And I was going to try and show you a real virus. Let me just do this little bit and we'll take the break 5 minutes late. Is that okay? Okay. 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 So here is adenovirus. Here is herpes virus. It's an envelope virus. The envelope has been removed. It's been a special method to look at it in electron microscopy where it's not stained, because staining changes the structure of viruses. And not only has it not been stained, because it's not stained, you can't see it very well in the electron microscope. So it's used some complicated software to overlay about in this case about 100 particles, one on top of another, so that every signal is reinforced if there's a signal in the same position in every virus particle. When you do that, if anything's floppy, it doesn't show, because in every virus particle it would be in a different position. So you only see what's in the same position in every virus particle. So the fact that you can see this much detail means that this is a very rigid virus particle, because you only see stuff that was present in all those virus particles. Okay. So what we have here, this is a real virus. This is, as I say, made from real viruses. And what we have here is, if we look at this subunit here, if you probably can't see from there, but if I look at it, it looks like it's got 6 corners. It's a hexon. You can see a little bit of the trace of the 6 subunits that make that up. And it's surrounded by 6, and that's 1, 2, 3, 4, 5, 6. So this has 6-fold symmetry. If we look at this one, if I can stop it there, we can see a penton. Let's see if this one will behave better. Yeah, this one will behave better. So here we have a penton. If we look at this one, it's got 5 little points, and it's got 5-fold symmetry. It's interacting with 1, 2, 3, 4, 5 of these. So this is what the inner capsid of a herpes virus looks like if you remove the membrane. Inside that is the nucleic acid. These holes are in the center of each of the hexons. They don't actually go right through the capsid. They stop below. And in fact, what happens is the subunits here have sort of got little towers on top, but down the bottom, they've got kind of a apron, parking lot or whatever, so that there's no gaps in the shell. They meet together at the bottom at their bases. You can't get through here. So this is what a real virus particle looks like. Okay. And one thing you'll notice, let me just quickly go through this. So what you find is polypeptides come together to form these capsimers. They interact tightly together. You see the pentons, you see the hexons. Those are the subunits that when you treat the virus with something nasty, it tends to fall apart. First of all, the capsimers come apart, and then finally you can dissociate the peptides in the capsimers. But the capsimers tend to look like capsimers because they've got much stronger interactions within the capsimer than between the capsimers. The capsimers have 2 types. At the 12 vertices, they have 5 neighbors, and so they're called pentons, so there'll be 12 of those. And on the faces, they have 6 neighbors, and so they're called hexons, and they will be some multiple of 20. You can have 0 hexons, or you can have a large number. Okay, when I said to you that the virus, when I showed you that herpes virus, I said to you it had been treated with a special technique so that it wasn't distorted when you looked at it in the electron micrograph. Normally, viruses were looked at with all these sort of nasty osmium tetroxide stains and one thing and another, and they were really badly treated by the microscopists, and they shrank. They became dehydrated. So this is what the model has always been, because it looks like an icosahedral nucleic acid in the microscope, and this is what they look like if you treat them with negative stain in the microscope. But if you don't, instead of having to spend a fortune on molecules that fall apart after medical students have played with them, and faculty, by the way, actually, if this was hydrated, it would become like this. And so a soccer ball is actually a much cheaper and better molecule of a virus than is this. And that's hardly like, you wouldn't really expect something that's trying to protect its nucleic acid to have lots of sharp edges and pointy things, which would be a really sort of place where you'd think it would be a really weak link where things could get in. So actually, A virus particle, when it's fully hydrated and feeling happy and healthy, looks like a soccer ball. And if you look at this soccer ball, what was garnet on here has now become fluorescent lime. It is fluorescent if I switch off the lights. And the hexons are blue. And a soccer ball has got 20 hexons and 12 pentons. Or at least they did till they started going really trendy on soccer balls recently. But this is the old-fashioned soccer ball. And that is the best model I know of for a virus. So it's just an easy way to take small pieces, put them together, and make a nice, firm, solid whole. And in this case, the nucleic acid will be inside. So thank you. Any questions? Okay, well, let's take our 10-minute break, come back about 8 minutes after.